It's a quite huge topic in human nutrition. It's impossible, I think, in one lecture to explain all the details about vitamins, so we're not going to do that. What I'm going to do is I will pick out the most relevant vitamins in human nutrition, so the ones for which we have deficiencies, as well as the ones for which we have excess and toxicities, and these ones will be discussed. So if we don't discuss a vitamin, it doesn't mean that it is not there, but it means that it is not the primary cause of concern today in human nutrition. You might think a little bit yourself, which types of vitamins are that? Because you have so many vitamins today. Well, if you listen a little bit careful or know already from literature, then the most important vitamins we're talking about is first vitamin A, which is mainly a problem in developing countries in the south, and mainly a problem for children younger than 5 years old. So you will see the mechanism on what happens with deficiency of vitamin A, and you will also see the different interventions that today take place in order to solve vitamin A deficiency. The second vitamin that is important, which is closely linked to vitamin A because the molecular mechanism is quite general, is in this case vitamin D. It's also a fat-soluble vitamin, just the same as vitamin A, and it plays a role, especially in elderly, because in elderly, they don't come out in the sunlight too much, and they also do not have very efficient conversions. Think about the term bioconversion. We discussed it already a couple of times. So vitamin D has a very strong bioconversion in the human body. And this conversion process is stimulated by UV radiation. But for elderly, the conversion process is not taking place that well. Also, they don't come outside in the sunlight too much. So that gives a higher risk of vitamin D deficiency. Also, in young children, you will have cases of vitamin D deficiency, which is also linked to a non-adapted dietary pattern that we will discuss later. Those two vitamins, A and D, are hormonal vitamins. So they directly interact with the genes and cause a nutrigenomic effect. The second set of vitamins that are important are folic acid, folate, which is actually vitamin B9, or vitamin M. And so these terminologies are not used that much. We usually talk about folate or folic acid. The target group for folic acid are pregnant women or women that would like to become pregnant. And the last one is very important, women that would like to become pregnant. So in fact, you should already have an intervention with folic acid before pregnancy starts. Because if you wait too long, actually the effect of folate is already gone. So we will talk about folate. We will explain the mechanism so that you understand a little bit why folate is important. And folate closely collaborates with vitamin B12, cobalamin. And cobalamin is also a risk vitamin, especially for vegetarians and veganists, because vitamin B12 is an animal-based vitamin. It is present in milk, it can be present in meat, fish, but it's not present in plants. So for people who only eat plant-based foods, you will not get enough B12. There is an option to avoid it, for example, if you would ferment the food, because bacteria are responsible or can be responsible for B12 synthesis. So if you would take plant-based foods and you would ferment them, then you might still have amounts of B12 in the final fermented product. But classical plant-based foods without processing are normally not rich in B12. So these are the four most important ones: vitamin A, vitamin D, folic acid, and vitamin B12. And in addition to that, we will have 3 vitamins which we will just briefly discuss because they might be relevant for some people in our society, but not for all. This is vitamin C and vitamin E. not because of deficiencies, but mainly because of toxicities, for example, by the use of supplements. And the final one is vitamin K, which can also be produced by the colon. But if you have people that have disturbances in colon function, or very young children where the microbiota is not yet fully developed, in those situations, vitamin K, can also be a problem. So that's more or less the program for the vitamins. So let's look at some general aspects. Vitamins life. And if you were a nutritionist in the 90s up to 1950, you would probably be working on vitamins because 11 Nobel Prizes have been awarded to vitamin research between 1928 and 1967. All the chemical structures that are linked to the vitamins have been discovered in that period of life. It was very fascinating because it was also the first time that people started to realize that in foods there are components that are essential for life. If you don't have them in the foods, you don't take them in, you get deficiencies and disturbances of metabolism. Now we know the vitamins very well. We know that they are organic compounds. They are not or insufficiently synthesized by the body. They're present in variable amounts, usually quite small amounts, and we need them for normal functioning at certain levels. We could say limited quantities, but it all depends a little bit on the situation. And if they're not there, you get very specific deficiencies. The remarks for that is that we have fatty acids, essential fatty acids and amino acids. These ones are not considered as vitamins. They're very closely related, but you have to think yourself, why do you think that essential fatty acids and essential amino acids, both of them were discussed in chapter 4 of the course notes, why do you think that these ones are not the same as vitamins? All of them are essential, the vitamins as well as these groups, but technically from a metabolic point of view, they're not the same. So try to think a little bit, where is the difference? The vitamins can be synthesized by the microbiota, especially vitamin K and vitamin B12. And if you have a normal microbiota, you don't need to take in additional vitamin K because the K is synthesized for you by the microbiota. For vitamin B12, that is not the case. We could synthesize it in the colon, but there's no guarantee that this B12 becomes available for humans. So that's why B12 has to be taken from the diet. And if you don't take animal-based foods, you have to take it from supplements, fortified foods, or processed foods, or animal foods. We see that the synthesis of vitamins and the capacity to synthesize can differ. And think about humans. Vitamin C is a problem for us, but for dogs and cats, it's not because they just synthesize vitamin C. And vitamin D is actually a vitamin because we need to activate it. And the activation is done by sunlight. So if you don't come in the sunlight or you're continuously in the dark, you might get a problem with vitamin D, or you have to take extra supplements to correct for that. I already explained the relationship between cobalt and vitamin B12, and we talked about that when we were talking about the trace elements. And deficiencies, using them as a parameter for trace elements deficiency is a little bit difficult because you could get some variable effects. And you will see that when we're talking about the effects. And another difficulty is that once you see a clinical deficiency, you're probably already too late. And so we should try to interfere a little bit more earlier so that the bigger effect, let's say, the clinical effect, will not be shown in later life. So what are general characteristics? Important to know is that vitamins are coenzymes. So it means that they are a cofactor for circulating enzymes. And they can actually help in the conversion from active to non-active forms, which is actually also a way by which we can measure the vitamin status in humans. And for example, for B12, B6, B1, B2, you can have an interaction between the enzyme and the vitamin. And it's strong but not covalently bound. So you can take it off in case you have a low amount of vitamin in the body. And that's why you get two forms, which is the hollow form, which contains the cofactor, and the Apel form, which doesn't contain it. And so you just take a blood sample, you analyze the proportion between the hollow and the Apel form. If the value is high, then you can say that the vitamin status is appropriate. If it is low, so if you have high Apels and low hollows, in that case, there are free circulating enzymes without vitamins, and then probably the vitamin status will be low. If it's a very covalently bound or a very weakly bound interaction, then you cannot use it, because then the link between vitamin status and hollow Apel form is not there. So it's only a little bit the intermediate situation of interacting between the enzyme and the vitamin. The last two conclusions on the slides are very important. We know that several vitamins, and I will give you the examples next week, several vitamins show conversion, so bioconversion. So it means that some compounds come in, but these compounds are not the active forms. You need to transform them into an activated form, which then causes the physiological effect. A good example is vitamin A and vitamin D. Both of them enter in the human body. They are then activated, and the activated form will interact with the genetic material to cause specific physiological effects. If you don't have the activation, you don't have the physiological effects. You will see later very good examples of that. A second thing to remember is that all the vitamins need to be transported and absorbed by the use of proteins. And these are specific proteins, so they are specifically targeting these types of vitamins. So if a person is malnourished and you get a serious form of undernutrition, for example, a protein deficiency, you can also have a vitamin deficiency. Not because the vitamin was not there, but because the transporters that are responsible for the absorption and the transport of the vitamin are physically not there. And in those situations, there's no transport possible, so you also get a deficiency at the level of the cells of vitamins. From a chemical point of view, we know that vitamins can have vitamers, which means that you have different chemical forms of the vitamins that are very closely related, like for example for vitamin E. Or sometimes you also have so-called provitamins, which means that these compounds are not that very active, but you need to convert them into the right activated form. A good example is a carotenoid, which is a colored substance that you can find in plant-based foods. For example, the orange color of carrots. This is beta-carotene. This one contains provitamin A activity. So if you eat the carrot, the carotenoid is released. It can be converted into activated vitamin A by bioconversion, and then you get vitamin A. If the bioconversion doesn't work, then you have far less vitamin activity after absorption of these plant-based foods. To know carotenoids, they are present in many plant-based foods. For example, if you take spinach, I think everybody knows that vegetable. A spinach is very, very green in color. But if I would remove the spinach by chemical extraction, then in those conditions, the spinach becomes red-yellow. And this red-yellow color or orange color is linked to the carotenoids. So they're physically there, but you don't see them as long as the chlorophyll, which is the green color, is dominating. If you remove the green color, all these carotenoid colors come up. So it means that they're present in quite a lot of plant-based foods. And some of them, not all, but some of them could have vitamin A activity. If we look to the classification, then we can actually say that traditionally speaking for vitamins, we always had a classification according to solubility. We had the fat-soluble vitamins and the water-soluble vitamins. All the fat-soluble vitamins are important. So we call them the ADIK vitamins, A, D, E, and K. All of them have problems, either with overnutrition, toxicity, or undernutrition deficiency. For the water-soluble vitamins, that's not the case. So some of them are a little bit less relevant. But still, there are several water-soluble vitamins that are important today. I'm thinking about B12. We're thinking about vitamin C. And we're thinking about vitamin M, which is folic acid. And so all of these are water-soluble vitamins, but still important in human nutrition today. And the requirements are known. Unfortunately, we started realizing the requirements based on studies that were done in the past with supplements, whereby toxicity effects occur. So we now know exactly what could be the maximum level of intake and which we should not cross in order to avoid toxic effects in humans. Requirements can be different according to specific conditions. I think that is linked to what we already said before. And these vitamins are in the text that you can find in chapter 7, classified according to activity. So we will first discuss the hormones, which is vitamin A and D. Then we will discuss the monocarbon transfer, so the methyl monocarbon transfer reactions, which is important for vitamin M and vitamin B12. So folic acid in combination with B12. And we could also use that to explain the effects of vitamin K. Vitamin H will not be discussed. It's biotin. It could be relevant in some conditions, but the other vitamins that we discuss are more important than that one. You have a whole cluster of vitamins that are linked to energy metabolism. Think about B1, B2, B3, B5. But generally speaking, some of them are only relevant because of processing. And generally speaking, we will not pay too much attention to that today, because if you optimize processing, in principle, there will be no deficiency. And for the antioxidants, we will discuss very shortly, but we will, because vitamin C and vitamin E can be toxic at high dosing. And it seems that several people like to take supplements, sometimes supplements in higher dosing, and this can give toxic effects in humans. That's why we need to talk about it, explain to you the mechanism, so that you are also aware that there are toxic limits in the intake of vitamins. And then finally, the deficiency. This is very typical for specific vitamins. The most important that we know is xerophthalmia, which is a deficiency that can occur in children. And this is an irreversible eye deformation. So these children who are younger than 5 years old can have irreversible damage of the cornea, which leads to irreversible blindness. So that's why there is so much attention to vitamin A, because we have to treat it at very young age, and we have to work in a preventive manner. So it means distribution of tablets in order to avoid that deficiency occurs. And I think there's a lot of discussion about it, because we need to find ways to make the interventions more sustainable. And so getting away from the tablets and moving on with traditional foods that are rich in vitamin A and that can prevent the formation of xerophthalmia. The second one is B1, not important, provided that technology is optimized. So we know that, for example, rice is a staple, especially in Asia. But if you eat only white rice, then the outer layers, the so-called aileron layers, are removed, which automatically also removes the B1 vitamin, which is located in the outer layers. So if you would like to have enough B1 intake, you have two options. The first one is to eat brown rice, because there the outer layer is still present and the vitamin B1 too. The second option is to adapt the technology so that you force, by high pressure, the vitamin B1 from the outer layers to the inner layers before you remove the outer layers. And so you can produce white rice rich in B1, provided that you force it from the outer layers to the inner layers. If that is not the case and rice is your staple food, then you get the so-called beriberi effects. There's a dry effect or a wet effect. It means you get weakness, stiffness and cramps, degeneration of the nervous tissue, while for wet beriberi, there is also transport of water, which goes subcutaneously and which gives you edema. So let's say accumulation of water subcutaneously, which means that also the blood will become very viscous. The heart has to pump and has to pump a lot of energy in it to pump around the blood, and this will give you the hypertrophy of the heart. So the heart will pump up, will blow up due to the enormous amount of energy that it has to supply to move around the blood. Then vitamin B12 and folate, which we will also discuss, mainly lead to formation of anemia. So I hope you remember what we discussed with the trace elements. I told you that anemia could be caused by copper and iron, but here I have to say that it can also be caused by B12 and folate. So you have 4 different components, nutrients, that can lead to anemia in humans. The technique is fast to do. It's just one small blood drop into a portable device, and you can immediately measure the degree of anemia. But the interpretation of it is much more complex, because you have several options that can cause the effect. And especially when you are in a malaria endemic region, also malaria can have an effect on the anemia process. So you have already 5 factors involved here. So what do you have to do in order to distinguish them? For malaria, it's very clear. You just need a microscope to look if the parasite is there or not. For the 4 other ones, you can divide them into the trace elements on the one side and the vitamins on the other side by looking at the precursor cells for red blood cells, which can also be done under the microscope. And so one precursor cell is called a megaloblast, which is a bigger cell that is the precursor for the red blood cells. If that accumulates in the blood, then you probably have a vitamin B12 or a folate deficiency, not a trace element deficiency. If you have a microcyte anemia, then it means that a very small cell will accumulate before the formation of red blood cells, and then you probably have an iron or a copper deficiency. Yeah, so by just looking under the microscope and discovering the precursor cells, you can already distinguish between the tracer elements and the vitamins. But to distinguish within the vitamins or within the trace elements, you need to have extra analysis. And based on metabolism, if you look carefully to what we have there, types of proteins, for example, you can already imagine which type of analysis you have to do in order to really know exactly which nutrient is responsible. And then vitamin C, scurvy. We already discussed that, I think, in the first lecture of this course. And most of the effects are neurologic and can give disturbances in the normal skin functionality. So you get dermatosis, which are some types of skin diseases that are typical for vitamins. Okay, I think with this, we have finished the lecture for today. So I will stop the recording